All right, we are here to talk about the process of mitosis and to sort of see how eukaryotes asexually reproduce. When we're talking about the eukaryotic and the life cycle for any cell, the cell cycle is going to be the entire lifespan of the cell. And usually we'd say that the life cycle starts at G1 right after the cell's divided before, and it goes up until that cell goes all the way through everything. G1, where it's doing normal cell processes, the S phase, where the DNA is going to get copied, and G2, the cell is going to get prepared for mitosis by making more organelles, fixing all the plasma membrane dings, getting all the cytoplasm, any enzymes that need to be made, and then the actual mitotic phase, which is when the division happens, and then it divides again. And then the two new daughter cells would then start their lifespan, or their cell cycle. In eukaryotes, the basic cell cycle goes interphase, M phase is short for mitosis, and then cytokinesis is the actual splitting of the cells, and the cell cycle is going to restart again after that. So when we look at interphase, there's three basic phases. Most of the cell life is spent in the G1 phase. G1 is simply normal cell functioning. The cell is basically doing everything it's supposed to do. It's making the proteins it's supposed to make and just doing the basic jobs that it does to stay alive. You have some cells in your body that might never leave the G1 phase, and that's completely okay. But once they get a specific signal that's reached to them, then that cell is going to move out of the G1 phase into the S phase. And once it makes that transition from G1 to S, it's kind of like pulling a trigger on a loaded gun. A bullet's going to come out of the barrel of the gun once you do that. That's sort of what happens here. Once you pass from G1 to S, then that cell is going to go all the way through, and it's going to go through mitosis, and two new daughter cells are going to start their life cycle at G1. So during the S phase, the DNA has to get copied, and that's kind of a big deal because when we make two daughter cells, each of those cells needs all the instructions. Think of the DNA as the blueprints for a house, and if you were going to build a house, you would want the entire blueprint. You'd want to know how to make the foundation, how to make the walls, how to put all the supports in, and you'd want all the right tools. The DNA provides that, so the two daughter cells each need the exact same copy of the DNA. That copying takes place in that S phase. During G2, the cell starts making more preparations for mitosis, makes more enzymes, makes more proteins, makes more organelles that are going to be divided among the two new daughter cells so that they have enough stuff to get going and perform all the functions that they're used to. So when we're talking about mitosis, we talk about one single cell dividing asexually into two new daughter cells. And what we have to understand is that when you're visualizing the phases of mitosis, the DNA is what we're really visualizing. During interphase, the DNA is in the form of chromatin. Chromatin's not really visible. It's kind of when DNA is just really long and stringy and kind of a hot mess all over the place in the nucleus. I always put this picture of the spaghetti up there because spaghetti is just long stringy noodles that are all over the place. You can't really see where one is and another one starts. You just see a plate full of noodles, and that's sort of what I picture inside the nucleus when the cell's in interphase. And mostly it's in G1. Once it gets a signal again, it's going to go to S phase and copy, and then move to G2 and get prepared for the actual mitotic phases. So let's talk about the individual mitotic phases, or the phases of mitosis. The first phase is prophase, and during prophase, a couple of huge things happen. First, that chromatin, that big plate of spaghetti, that chromatin is going to condense, and it's going to form the more familiar chromosome that we're used to seeing. As the chromatin's condensing, remember that the DNA got copied during the S phase. Those two exact copies are going to get joined together, and they get joined together at something called a centromere, and this sort of gives kind of this X shape to the actual chromosome. We can sort of see that DNA is really tightly wound together and being held together at that centromere. Each side of that chromosome, each individual copy of the DNA that should be exactly the same because the cell copied it during the S phase, is going to be called a sister chromatid, and the entire thing is called a chromosome. On that centromere, which is holding the two sister chromatids together, there's something called a kinetochore on each side, and the kinetochore is what's going to be attached to so that the DNA can be moved around the cell and divided equally. The things that are going to be making the DNA move are something called centrosomes, and centrosomes are little centers that are filled with the organelle, the centrioles. The centrioles are outside the nucleus, and they really don't do anything at all in the cell's life cycle until it's time to divide. And once the cell hits prophase, those centrioles migrate to opposite poles of the cell. So you have one centrosome on one side, one centrosome on the other side. And those centrosomes start creating spindle fibers, which are microtubules, which is more of the protein cytoskeleton filaments that can be used to help move the DNA. Those spindle fibers are going to attach to the kinetochores on the DNA, and the centrosomes are going to use that to sort of move things around. I always kind of think of the centrosome as kind of like a fishing rod and reel, and the spindle fibers are kind of like the line that comes out. And that spindle fiber is going to attach to that kinetochore and be used to move the DNA all around. As the cell is transitioning from prophase to metaphase, some books will tell you that there's a phase called prometaphase, which is kind of like that combination of the two. During prometaphase, the nuclear envelope is going to break down, and that's fine. Usually DNA has to stay inside the nucleus because it's really important. It can never leave. But if we're going to divide the cell and we need equal DNA on both new daughter cells, we're going to have it leave the nucleus at some point. So that nuclear envelope is going to break down and allow the DNA to be moved. The next phase coming up is going to be metaphase, and when I see meta, I think of the middle, and chromosomes are lined up along the cell equator or the middle of the cell. Remember, we have those two centrosomes at either end of the cell, and they have the spindle fibers attached to the kinetochores of the chromosomes. Well, they're going to move those the chromosomes, to the equator of the cell and just sort of line everything up. And as soon as it's all lined up, then the cell's going to transition to what's called anaphase. In anaphase, big stuff happens. First, that centromere that was holding the chromosome together is going to disintegrate, and each sister chromatid is going to be pulled by the centrosome to opposite ends of the cell. Now, if you remember, the sister chromatids had exact copies of the DNA, and this is a very active process, so a lot of ATP is used, and that ATP came from cellular respiration that we were talking about in earlier lectures. So the ATP gets used to help pull these sister chromatids to opposite ends, and since they're getting pulled to opposite ends, that ensures that each new daughter cell is going to have an exact copy of the same DNA. After anaphase comes telophase. During telophase, this occurs once all the DNA is on either pole of the original cell, and all of a sudden nuclear envelopes are going to start forming around that DNA. Remember, that's really important stuff in the DNA, so it's a kind of a big deal that we put it back in a nucleus and sort of seal it up and keep it in one place. Once the nucleus gets sealed, those chromosomes that were nice and condensed from prophase at the beginning of mitosis are going to unwind and turn back into chromatin. As all that's happening, the spindle fibers are no longer needed, so they disappear. The centrosome is going to disappear. Once we get to the end of telophase, then the process of cytokinesis is going to begin. And in animal cells, there's something called a contractile ring that's used, which the contractile ring kind of pinches the plasma membrane in towards itself and creates a cleavage furrow until both ends of the plasma membrane sort of meet, and then they have a slight separation, and you have two new daughter cells. Plant cells have a little different process. Plant cells create a cell plate, which is just kind of like some cellulose fibers sort of getting in there in between the two new nuclei that were formed and creating a new cell wall, because plants have to have that cell wall in there, so they need something a little bit more heavy duty than animal cells that use the contractile ring. Now, once all this is done, this is huge stuff right here, is that we get two diploid daughter cells that are genetically identical to each other in the parent, which means the same exact DNA that came from that original cell that really doesn't exist anymore in the being that it once was. Now, that same copy of that cell, there's two daughter cells, and those two daughter cells are exactly the same. If they're not exactly the same, they're not going to work the right way, and that's could cause some kind of problems later on down the line with the cell's functioning. It might not do the things it's supposed to do. And hopefully we know that our bodies like to be pretty efficient and our organisms like to be pretty efficient. So not doing what you're supposed to do is going to be the exact opposite of efficient and could cause problems for the organism as a whole. Here's a picture that sort of shows how the contractile ring sort of pinches the sides of the cell back in on each other. And this right here, that little divot that's on either side, that's what we call the cleavage furrow. And that's what you can see in this real picture of what's going on. In the plant cell, we can see that the cell plate starts to form and eventually creates that cell wall between the two new nuclei. And again, two identical diploid daughter cells. They're diploid because they have two copies of the DNA in there. One copy from this organism's male parent and one copy from the organism's female parent. So if we look at all this again, let's just sort of look at these. On the top, these pictures are from a fluorescent microscope. I'm not a big fan of those pictures, but these drawings on the bottom are pretty good. So I'm going to sort of point to these and focus on these as I sort of recap these steps. First, in interphase, the DNA isn't really visible. It's in the form of chromatin, so it's just all over the place. We can see the centrosomes with the centrioles are just sort of sitting outside the nucleus and the cytoplasm, just sort of hanging out doing their thing. Once the cell's gotten the signal to move forward, the DNA is going to copy and it's going to prepare for mitosis, and once it gets into prophase, then that chromatin is going to start condensing. And it starts condensing into little strings at first. The centrosomes are starting to migrate to the poles, and then the sister chromatids, which are the exact copies of the same DNA, start to form, and you can start to see that classic X pattern. As we move from prophase into metaphase, that nuclear envelope's got to dissolve so the spindle fibers from the centrosome can attach to the kinetochores on the centromere of the chromosomes. After all the DNA is sort of attached to the spindle fibers, which is attached to the centrosome, everything gets lined up on the cell equator during metaphase. During anaphase, the centromere breaks down and sister chromatids are pulled to opposite ends of the cell. And finally, in telophase, the nucleus starts to reappear on each new daughter cell, and the contractile ring starts a process of cytokinesis where the cell basically divides all of the parts and everything going on. Well, how is all this stuff regulated? Well, asexual reproduction in prokaryotes, that happens whenever the conditions are right. If the prokaryotes have food, they're at the right temperature, they're going to divide. That's why we put food in a refrigerator, because once you get it below down to that like 36 degree Fahrenheit-ish kind of area, or about 4 degrees Celsius, then bacteria don't divide. So it keeps our food fresher longer, and we don't have to worry about foodborne illnesses. Eukaryotes, we don't have this freedom. We have to get a signal. And one of the places we can get the signal in eukaryotes is something called growth factor. One place where we can get growth factor is from platelets. Well, platelets are cells that are in our blood that help clot the blood so that we don't bleed out if we have a cut. If you ever cut your finger, it'll stop bleeding after a couple minutes. That's because platelets go to the area and they sort of form this blood clot, a good blood clot I like to say, because there's bad blood clots that can kill you. This one's a good one because it stops you from bleeding. Well, as platelets go to area where you have damaged cells, they're going to release growth factor. And hopefully that makes sense, that you have damaged cells that need to be repaired. So they tell cells that are in the area to, hey, let's start dividing. Well, that regulation happens in the G1 phase. If there's no growth factor released near the cell, nothing happens. Once it passes the restriction point, though, that's when I said that it goes from G1 to S. That means the entire cell cycle is going to happen, where it's going to go S and copy the DNA, it's going to go to G2 and prepare for mitosis, and it's going to go to mitosis and create two new daughter cells. The thing that sort of gives the message to move forward from growth factor is something called a cyclin. And cyclins bind with something called protein kinase, and hopefully by the name of that word, protein kinase, you can tell that that is an enzyme with the ASE at the end. The specific kind of protein kinase we're talking about is cyclin-dependent kinase, or CDK. Well, CDK is inactive, or the active site is closed, if cyclin's not around. Once cyclin gets activated by growth factor, well, the cyclin's going to attach to that CDK, and it's going to open up the active site. If you remember back from our protein talks a long time ago, when enzymes are regulated by the attachment of a molecule somewhere other than the active site, that's called allosteric regulation. And that's basically what's going on here. So let's look at a little diagram and sort of see the sequence of events. The first thing that's going to happen is growth factor is going to be released. That could happen from something like platelets because you injured your skin. Next thing that's happened, growth factor stimulates the synthesis of cyclin. Cyclin gets put together, and cyclin is going to act as that allosteric regulator to turn on the CDK. So when cyclin is synthesized, CDK gets activated, and once CDK gets activated, that creates a cascade of cell cycle events, going from S phase to G2 through mitosis and cytokinesis. If we look at that on this picture, we can sort of see that happening. Look at how we see CDK in every phase of the cell cycle. It's all over the place. The only time CDK looks different is when there's cyclin attached to it. Well, the cyclin only begins to synthesize if the growth factor is released. Growth factor tells cyclin, hey, start to get put together. When the cyclin attaches to CDK, look at the difference. Now we have that little active site going there, and that's going to catalyze reactions to push the cell past this restriction point. Otherwise, the cell's just going to stay here forever if there's no cyclin present. Once it goes past the restriction point, the cyclin actually breaks down and just sort of gets absorbed back into the cytoplasm. But since the restriction point has been passed, the cell's going to go all the way through the cell cycle until there's two new identical daughter cells. Here's one more look at what's going on. There's interphase again. Remember, we have chromatin, which is the form of DNA in interphase. During prophase, that chromatin is going to condense into chromosomes. During metaphase, those chromosomes get lined up along the equator of the cell. The centrosomes are at each pole of the cell, and they're going to pull the DNA apart. In anaphase, the centromere has to split first, and then each sister chromatid gets pulled to opposite ends of the cell. Finally, in telophase, two new nuclei are formed around there, and you're going to end up with two new daughter cells. This is a really important process that helps to make sure organisms sort of doing things the right way and working with healthy cells at all times. Next thing we'll be talking about on this series is going to be meiosis. Hopefully you enjoyed this. Have a good evening.